Across the living world, phosphorus moves through green leaves, bones, seeds, and soil. It helps cells store energy, build membranes, and pass inheritance onward through generations. Yet in many plants, phosphorus is packed away behind a quiet lock. That lock is phytate, a mineral vault built inside ripening grain and seed. Wheat bran, rice bran, corn, beans, lentils, and oilseeds all carry it. In the seed, phytate is not a problem at all, it is planning. It is a savings account for spring, waiting patiently beneath the husk. When rain arrives, those savings help a pale new root begin life. But to many animals, the stored nutrients are surprisingly hard to reach. Phosphorus, iron, zinc, and calcium can remain tied within the sturdy molecule. For farmers, this locked abundance once meant waste and extra mineral inputs. For rivers, it could mean excess phosphorus drifting from fields into downstream water. Long before laboratories, nature had already found the missing key in living tissue. When a seed germinates, its own phytase wakes in the dark. The enzyme loosens stored phosphorus for the embryo's first tender push upward. Soil fungi and bacteria also make phytase around living roots after rain. In the rumen, microbes help grazing animals open green plant stores quietly. Every forest floor is partly a quiet phytase workshop after steady rain. The human story began with seeds examined closely under lamplight and glass. In the 1850s, Theodor Hartig studied mineral deposits in plant tissues. He described a phosphorus-rich substance gathered inside resting seeds and dormant cereal grains. Later scientists called it phytin, and then phytic acid in plant chemistry. They knew the treasure was present, but not the biological opener. In early food chemistry, bran behaved like a stubborn mineral reserve. Minerals appeared in measurements, yet were not always available in living bodies. The missing actor would emerge from rice-bran in early 20th century Tokyo. In 1907, Umetaro Suzuki studied an unusual rice-bran activity. With Koji Yoshimura and Mitsugi Takaishi, he named the enzyme phytase itself. Their extracts split phytin and released phosphate into clear solution. A new enzyme had stepped from agriculture into the language of chemistry. Suzuki later became famous for discoveries that helped define vitamins in nutrition. But here, he helped reveal a seed's hidden biochemical locksmith at work. Through the 20th century, phytase appeared in many living places and fermentations. Researchers found it in grains, yeasts, molds, bacteria, and digestive microbes. Each discovery made the same quiet lesson feel larger and more practical. Wherever plants stored phosphorus, other life learned to recover it again. Industrial interest grew around one practical agricultural question in animal nutrition. Could an enzyme unlock feed before nutrients passed unused through animals? By mid-century, microbial phytase was being tested with farm animals. Studies showed more plant phosphorus could be used by poultry and pigs. Less mineral phosphorus needed to be brought in from outside sources. Less phosphorus could leave farms in manure, ponds, ditches, and runoff. The industrial origin story gathered strength inside carefully tended fermentation tanks. Microbial strains, especially Aspergillus niger, became important enzyme sources for scale. By the early 1990s, feed phytase entered routine agricultural commerce. A discovery from rice bran had become a tool for modern farms. To understand its grace, imagine phytate as a small ringed scaffold. Around that scaffold hang phosphate groups like tightly fastened parcels. Phytase settles around one parcel and draws a water molecule close. A bond opens and a small phosphate piece drifts free into water. Then the enzyme lets go, unchanged, ready for another bond. Step by step, the vault becomes less tightly sealed and more available. The released phosphate can nourish cells, bones, leaves, or microbes. The remaining inositol frame continues through ordinary biological pathways inside cells. Nothing here is violent. It is patient molecular housekeeping at seed scale. In animal feed, phytase helps grains give more of themselves to nutrition. It supports nutrition while easing pressure on mined phosphate rock. It can reduce phosphorus burden in barns, ponds, and watersheds. In aquaculture, it helps plant-based diets fit fish more gently. In whole grain baking, phytase can soften the mineral lock during fermentation. Fermenting dough becomes a small landscape of enzymatic release and rising bubbles. In brewing, phytase helps manage cereal minerals during fermentation. In plant-based foods, it improves access to hidden nutrients within seeds. In soil research, phytase points toward phosphorus cycling with less waste. It reminds us that chemistry can serve by careful subtraction, not excess. Less runoff, less mining, less locked nourishment, passing away unused. A small enzyme connects seed dormancy with the future of agriculture. Phytase leaves us with a quieter vision of progress and industry. Not larger extraction, but wiser release from what already exists around us. In that release, biology offers industry a greener tomorrow, patiently and persistently. Behind Enzymes Bio is a team of scientists who love this quiet biology. We gather enzymes from across the world so researchers and businesses can order from one home. Nature's technology for a greener tomorrow. See more at enzymes.bio.